Good morning, everybody. I am Drew Armstrong, and I'm your host of Post-Hoc Live. I'm also the executive editor over here at Endpoints News. Happy Halloween to those of you who are celebrating. I am very excited about our show today. It goes back to something that we have been talking about all year here at Endpoints. Um, Earlier this year, our science correspondent, Ryan Cross, came to me with a tip. He has been thinking and writing about CRISPR gene editing technology and was asking the very provocative question about when this technology was really going to make the huge impact that I think we all thought and hoped it would. He found out that a team of researchers in Philadelphia were doing something revolutionary. They were working their way toward treating single patients with unique mutations, designing boutique therapies that otherwise wouldn't exist. And I guess, as most of you know, that is the baby KJ story. And Ryan told the world how that works and that cure, if you could say that, came to be. Now, those researchers, University of Pennsylvania scientist Kiran Musunuru and Children's Hospital of Philadelphia pediatrician Rebecca Ahrens-Nicklas, are getting ready to take the next step and bring their technique to more patients, potentially a lot more. This morning, they published an article outlining those plans, and they are trying to make it easier for other scientists to follow in their footsteps by publishing their briefing books they submitted to the FDA for their pre-IND meetings, as well as the FDA's initial responses. They are going to join me and Ryan on the show today. We're going to talk about what they're doing, how it might break through one of the biggest bottlenecks in gene editing and rare disease. Now, a little bit of housekeeping. If you are joining us for the first time, hello, thanks for being with us. Post-Hoc Live brings you the reporting, the analysis, and the debate that usually happens here at Endpoints behind the scenes, live to you. If you are with us on YouTube and you're not already following us, please hit that little subscribe button below our feed. You get alerts about future live streams. You can also sign up for the Post-Hoc email on your Endpoints News subscription page. So Kiran, Rebecca, thank you so much for being with us. Ryan, great to have you back on the show. I want to Let's start here. Maybe you guys can walk us through the decision to publish this paper, what's in here, and how you all think that this is potentially going to help bring this technology and this approach to a lot more folks.
Sounds great. Thanks for having us this morning. Maybe Kiran, I'm happy to start with our thought process. So throughout this process, we know that there is a large gulf, I would say, between the world of developing a gene editing therapy in the lab and trying to actually advance it into clinical practice, not just into a first-in-human study or first-in-human experience, but actually try to scale this so it could be distributed to more patients if it's found to be safe. And the only way that we're going to sort of span that gulf is in partnership with our regulatory colleagues and with regulatory agencies. But a lot of times, especially those of us who are academic physician scientists, regulatory interactions are a big black box, right? We don't do this every day, and we don't have access to a lot of those documents that maybe people who are well seasoned in industry or other fields have. And so we thought it was really important to actually lay out what goes into even preparing or having a conversation with the FDA. And more importantly, in this case, what we've learned, because we know that things are changing incredibly quickly in terms of guidance and regulation around gene editing therapies. And so we just wanted to be very transparent and say, okay, this is what we asked, this is what we've learned. And we hope others not only learn from it, but can borrow from it. We're happy for people to share similar strategies, to use similar language if it's appropriate for their work, and also to provide us feedback, right? Somebody might have a great idea that we haven't thought of, and we would love to be able to learn from others. So we thought transparency was key, and we had the ability to do that because we're federally funded investigators. So we felt we had an obligation to share what we've learned. Kiran, I don't know if you want to add more.
Yeah, if I could add some color to that. So there's been a lot of talk from the FDA leadership, the previous administration and this administration, about the potential for gene editing to be a platform, to be something that could be deployed to many patients, to be personalized in a way that most other technologies cannot be personalized. And a lot of affirmation of this concept, but very little in the way of details. And that makes sense because the FDA is not going to weigh in on hypotheticals for the most part. What happens is that sponsors, whether they're industry sponsors or academic sponsors or sponsor investigators, as we would call them, actually have to bring something to the FDA, propose something, and then the FDA will consider it and then give their feedback, and then you move forward from there. So until very recently, it didn't seem like anyone had actually pushed the FDA on, okay, here's our proposal for what a platform looks like to take advantage of the properties of CRISPR gene editing. What do you think of this? Perhaps industry has done it. It's hard to say because, the propensity of industry is to keep things confidential, trade secrets, right? And so entirely understandable. But if you don't know what is happening behind the scenes, then that doesn't help anyone.
Yeah.
And so we felt that as academics, we had the opportunity to be the tip of the spear, so to speak, to actually approach the FDA with what we thought a platform should look like,
Hmm.
engage with the FDA, get feedback from the FDA. And we've actually done it twice now. The first instance was earlier this summer with the disease PKU, phenylketonuria, and then later in the summer, early fall, with a more ambitious platform encompassing 7 different urea cycle disorders. But in each case, we got specific feedback from the FDA and actually reached pretty good agreement with them as to what the phase 1, phase 2 clinical trials would look like, what the path is for to actually potentially eventually getting approvals, not for a single drug, but for a platform of drugs. And we felt we're not beholden to the same pressures as industry is. We're federally funded. In fact, there's an expectation that we share widely.
Hmm. Hmm.
And so that is exactly what we're doing. We felt it was time, and we had our pre-IND interaction for the second platform on urea cycle disorders just last month. And so as soon as that happened, and we realized, okay, we've made some real progress, and it's important that everyone knows about it. We started writing and preparing our documents and getting everything all together so that we could get them published. And today, this morning, happens to be the time when it all comes out.
Uh, can I ask you guys, I think what is probably a bit of a facile question and that you've implied the answer to in your comments here, but when CRISPR kind of was really rising to prominence or even hype. I mean, like I think a lot of us looked at these either N of one diseases or N of just a few diseases, in the ultra rare space. And we're thinking like, oh, this is a really obvious application of this. And like, great hope that like this would explode into all sorts of treatment for exactly these type of patients. And that really didn't happen until I think now. And, I'm wondering like just stepping way back, why is that? And why does it have to be you all that are doing this? Because I feel like there's so much in biotech and pharma where like the capitalist incentives and line up really, really well. And we see, great things happening that are helping patients and things like that. But this has been, I think, a bit of a mystery to me that, and maybe my expectations were too high, but like this felt like it should have been a thing that, technical challenges aside, like could or should have been happening a long time ago. Like, why not? Why did it, I'm glad you guys are doing this, but why did it have to be you all?
Yeah. So, to be fair to the companies in the gene editing space, if you turn back the clock 5 years when the biotech sector was booming, there was a lot of ambition. And if you looked at pipelines of the various companies, it was chock full of rare diseases, ultra rare diseases. There was very much the intention to apply these technologies to patients with unmet needs, to diseases where there were no alternative therapies. And then what happened over time is, it's all cyclical, of course, boom, bust, and so forth. And we're in a downturn, as we all appreciate right now, for a variety of reasons. And the companies became much more conservative. And you understand they have to answer to their investors, they have to answer to their boards. And so it just wasn't tenable for them to continue to have these pipelines full of ultra rare indications. And they were essentially in a situation where in order to raise more capital and keep things going, and they had to pivot to more common diseases, or at least the more common among the rare diseases. And that has led to, from our perspective as physician scientists, a somewhat unfortunate situation where all of the companies are gravitating towards the same few indications and in some sense over serving those particular indications, whether it's sickle cell disease or alpha-1 antitrypsin deficiency or hypercholesterolemia or what have you. And that leaves thousands and thousands of rare, ultra rare, even N of one type scenarios behind. And so again, as academics, we have the opportunity to be the tip of the spear because we're, in a sense, answerable to no one except ourselves, except to the extent that we need to be able to raise, NIH funding and, other sources of funding to do what we want to do. But once we have that funding, we have the ability to do effectively whatever we want. We can be impulsive in a way and say, we're going to go after something crazy and ambitious, like trying to help one newborn kid with an N of one scenario, a variant that's unique to them, a disease that is a 1 in a million, and actually try to commit the resources and the effort to make that therapy for that kid. And it was crazy.
I think I would
Looking
take it.
back, we're still trying to wrap our heads around it, but
Yeah, I mean, it's an
Yeah.
amazing thing
Go
you
ahead,
guys have
Becca.
done. Yeah.
Yeah, I mean, I think I would take it a step further and say we have the freedom to do this, but in some ways we also have the obligation to do this. And I think this is also the rare disease geneticist in me. But right now in the rare and ultra rare space, the people that are leading the charge for the development of novel therapeutics are often the families of our patients. And that's an incredible burden, not burden, but expectation, an incredible amount of work that's already being sort of shouldered by a group that has so many challenges in their life as it is. And so as the physician scientist, and in my case especially, that cares for these kids, I really think we have an obligation to try to help them because, as Kiran said, we have the freedom to do so. And so and this is a big driving motivating factor for why we released our documents, is we want to be able to help even more kids than the populations or the diagnoses that we have the ability to translate ourselves. Right. It's all about sort of growing this effort such that it can spread for a variety of rare and ultra rare diseases.
Maybe to help set the scene for why you think a bigger clinical trial is the way forward for this field. Maybe just tell us a bit about the feedback and reactions you got after publishing and presenting on the therapy that you made for baby KJ back in May. I understand that maybe reactions were a little mixed. There was a lot of praise, but maybe some people felt a bit differently.
Yeah, no, and appropriately so. I mean, I'm going to have to tell you, I felt a little bit conflicted about all of this, right? While it's very exciting to show proof of concept that hopefully this was safe and hopefully it provided some benefit to KJ, I care for so many patients, and there are so many rare and ultra rare patients out there that this was not a possibility for at this point in time, right? That it wasn't their child that had gone through this process. And so we knew that we were going to try to have to scale. And we knew that there was a lot of unmet need, but the feedback from the rare disease community was such that we knew we had to work quickly to make that happen. We have gotten thousands of requests for help. And we, just like we have an obligation to try to be transparent about our work, we have an obligation to try to help, I feel. And so we immediately started thinking through what that would look like. How do you actually scale? Because it was, as was appropriately pointed out by a lot of folks, a lot of people donated their time, their effort. The stars sort of aligned to make a case like KJ actually translate into the clinic.
Yeah.
But that's not sustainable. We need a sustainable model. And that's really what we're trying to build here. And at least in the US, a sustainable model in the rare disease space only comes once you have some sort of approval. And we know conditional approvals versus full approvals sort of warrant different insurance reimbursements and things along those lines. But at least some form of approval will allow us to start thinking about how we can sustainably manufacture and distribute these drugs. And so to get an approval, you need a formal clinical trial to show that a drug is safe and that it does something for the patient population. And so that's why Kiran and I have been sort of tunnel vision since this story broke, and really even before then, to figure out how we can get that trial off the ground as quickly as possible. I don't know, Kiran.
Something that was really that really stuck with me from our conversations earlier this year was, I think, Kiran, maybe you might have coined this phrase, mutational discrimination, right? You didn't want to say, we're just going to pick a few variants. Maybe they're the most common ones. Maybe they're the ones we happen to work on in our lab, and make CRISPR therapies for just those variants. That you wanted to just treat whatever patients were in front of you, regardless of what their mutation was, if you thought CRISPR could fix it. And I don't think your thinking today seems a little different from that, maybe a little evolved from that, not completely, but help me understand how what you're doing now in a clinical trial is different from the just treating whatever patient is in front of you.
Yeah, certainly. So to set the context and to get back what we were discussing earlier. So one very important reason that companies have feared to tread where we're treading in terms of going after N of one type scenarios is the prevailing model has been one where you spend years and invest a lot of resources, up to hundreds of millions of dollars, to make an optimal drug to help one particular group of patients, tackle one particular variant. And the natural question, given how companies are set up and who they're answering to, investors and boards, is how do you get the biggest return on that investment? And obviously, if you're going to be able to help 100,000 people or more with that single therapy into which you've invested so much effort, you're going to choose that over a single patient. It just, it's dollars and cents, and it makes sense, and you can't really criticize anyone for that. But it creates these incentives that lead to that situation that I've described, as you noted in the past, as mutational discrimination, where there's naturally going to be a bias towards high frequency variants and higher frequency diseases, and of course in high income countries rather than low and middle income countries. And so, our stand was we want to be able to help any patient who comes to us at the Children's Hospital Philadelphia, whatever flavor of disease, whatever particular gene is affected, whatever particular variant, and get each and every one of them a fair shot. And we were very happy that that is the way it played out with KJ. There was nothing special about KJ per se. I mean, he's a very special kid, and of course, holds the highest place in our heart. And he's incredibly adorable and happy, and
But it was right place, right time.
But he was there in the right place at the right time. But it wasn't anything about that particular gene, CPS1 versus OTC versus any of the other urea cycle genes, or his particular variant, compared to any of the myriad of other variants a patient like him might have. He was in the right place at the right time. We were ready, and we did it for him. And that was kind of the original vision. Any patient who presents locally to our hospital, we want to help them. And what changed, and I wouldn't say it's a total change in our perspective, but something of an evolution, is that prior to KJ's case, we were somewhat limited in our scope. We were thinking locally. We're thinking the patients who come to us.
Right.
After KJ, after the announcement, all of a sudden, what happened is the world became our catchment area, our constituency, right? Because we, as Becca said, we were getting thousands of requests from all over the world, far-flung countries, asking for help. And as Becca noted, the vast majority of them were not in a position to help yet because the nature of the disease is, we don't have the delivery technologies to, get to this, for example, the central nervous system and tackle the many, many neurodevelopmental disorders, or, various muscle disorders. We're not quite there yet. But, even there's a small fraction of the families that have reached out to us where the disease in question is one in the liver, one that we could tackle. And so it becomes much more complicated because being able to give each and every patient a chance now means anywhere, right? And we can't do one-ups. We can't do single patient expanded access IPs like we did with KJ. We quickly run out of resources, exhaust everyone involved in the process, and then it would all come to a screeching halt, and then we wouldn't have done much more than helped a handful of patients. Whereas setting up a clinical trial that is potentially open to anyone, anywhere, there are still some practical limitations in terms of, getting patients to where we are and being able to host them, and, the resources that have to be spent in that respect. But in principle, being able to have an open clinical trial that is accessible to any patient anywhere who meets the inclusion criteria, and in this case, with a urea cycle disorder platform, any of 7 different diseases with variants in any of 7 different genes who would qualify, they have a shot at this.
Maybe just to talk about some of the details of how this actually all plays out, and some of the things that you've proposed to the FDA to streamline this so that you're not redoing all the science, all the development for each new patient, each new mutation. In the detailed article you published this morning in the American Journal of Human Genetics, and some of the outlines of the interactions with the FDA, there were a couple details that really stood out to me. When one of them was, I think, the fairly bold request that you made to minimize the amount of preclinical work, minimize the amount of animal studies that you do, for each of these new variants. Can you maybe just walk us through that briefly, what you've proposed and how that differs from how drugs are normally developed, and what the FDA's response to that was?
Yeah, I'm happy to do that. I will say first and foremost, the thing that's front of mind is that we do this in a safe way. And so I think what we've outlined, in our opinion, really maximizes safety, while also being realistic about what you need to do to show that safety. And really what we asked for is that we do an entire what we call suite of IND enabling studies. So a really nice group of studies that show that a lipid nanoparticle delivered base editing drug is safe in animal models, right? And similar to what you would do for traditional drug development for the first drug product, for the first version of that gene editing drug, right? Because the safety of the drug is really dictated by the delivery mechanism in this case, or the lipid nanoparticle. The cargo inside, the base editor and the guide RNA, don't actually change the overall, at least acute safety toxicity profile that much. And so what we thought was that we could take that one set of studies and then leverage them over and over again. Because we know that those IND enabling studies are incredibly expensive. And often rare disease communities or researchers trying to develop this work just don't have the resources to be able to do this for every specific genetic variant that might cause a rare disease. But by being able to go back and cross-reference that suite of IND enabling studies, it's going to save not only resources, but time. And we know that time is of the essence as we're trying to develop these targeted therapies, especially for something like a urea cycle disorder. But on the flip side, yeah, go ahead.
Oh, I was actually just going to break in with a little bit of news that just crossed the wires. One of my former colleagues over at Bloomberg just has a story out. You guys may have been aware that this was coming. I guess Vinay Prasad has made some comments about saying that the FDA is going to be extremely flexible and work very fast with the scientists who want to bring these therapies to kids who need it. They're going to publish a paper next month outlining what they're calling a new approach to this. We're seeing, for those of you who care about stocks, a lot of CRISPR stocks are suddenly moving up very, very high. So I nice to have news on the show. This seems like good news to you guys. Were you guys aware that this was coming? I'm not sure if you can bring it. Sorry to cut you off, Rebecca, but
no,
anytime we've got news on the show, I love to. We're live, so we might as well get into it.
We are used to things changing in real time recently. I have to tell you, I feel like every day there's something new and exciting.
Yeah.
Kiran, I don't know if you want to answer.
Yeah, so we had heard rumors that this plausible mechanism pathway, as it's called, is coming. We've also heard that it was directly inspired in many ways by KJ's case, and the concept that at least had been outlined in what has been publicly reported, and I'll be curious to catch up with the breaking news this morning as it unfolds, is that with a relatively small number of subjects in a clinical trial. And again, this is intended for personalized therapies where subject number one receives one personalized version of the therapy, patient number two receives a different one that's tailored to them, and so on and so forth. With as few as 5 patients, if the basic drug is safe and if there are relatively consistent effects in terms of efficacy, that might be enough for the FDA to give serious consideration to a conditional approval. And that's absolutely breathtaking in terms of the possibilities for patients with, rare, ultra rare, N of 1 type scenarios. Entirely unprecedented. It would be transformational. I think we can all agree on that. And it actually dovetails very nicely with what we have been working with the FDA over the past few months under the more traditional model. And so with the urea cycle disorders, if you dive into the details of the American Journal of Human Genetics article and the linked documents, so from the FDA, the written feedback from the FDA, where we've received agreement from our colleagues at the FDA, is that we can do a phase 1/2 clinical trial, what we're calling an umbrella of umbrellas clinical trial, because it spans 7 different urea cycle disorders. And we can enroll 5 subjects spanning at least 3 of those 7 disorders. And if things look good after they're treated, we would be in a position to ask for an end of phase 2 meeting and talk about an extension to phase 3, which obviously would be a huge step forward, because that puts us in line with this, maybe 5 to 10 additional patients to actually get all the way to approval. But with this plausible mechanism pathway, we might be talking about an even more accelerated path to at least a conditional approval. And so that raises the interesting question, well, if we launch this urea cycle disorder trial then in the coming months, which is our fervent hope to do so, it'll depend on the FDA clearing our IND application, of course. Maybe in 1 to 2 years, we'll have actually enrolled enough subjects and treated enough subjects to be able to move to a conditional approval. And as Becca noted earlier, that is the key to make this financially viable. While the trial is ongoing, any resources you put in, they're sunk costs. You're not going to be able to get reimbursed for those costs, right? But once there is an approval, then you have a shot at convincing insurers, Medicaid, what have you, to reimburse for at least the cost of the drug, the cost of the development, hopefully, something on top of that. And then it becomes self-sustaining, becomes a virtuous cycle. Then you can scale it up and actually be able to serve the needs of all the kids in the country who need this kind of therapy. And it starts with urea cycle disorders, but there's no reason you can't replicate more widely for many other diseases.
And just to build on that, I think that if a new, pathway becomes available for some sort of accelerated approval that requires enrollment of fewer subjects, there's even a greater sort of responsibility of investigators to have utmost transparency, right? I think it's really, rare disease families, rare disease physicians are all very excited about the possibility of targeted therapies, but also you don't want the perception to be out there that you're moving so quickly that you're not doing this in a safe way. And so I will say we're very excited about this possibility, but it also presents a great response, or puts on investigators a great responsibility that you do this in such a way that you are transparent, that you are safe, that you're telling people how you're doing this, so you can build the trust of patients, of the communities, of funders, of everyone, that you're thinking through how to evaluate these drugs in a way that is both cost effective and resource effective, but also still maintains safety for families.
I know we started the show with Drew's question on sort of why is it taken so long for us to get to the point where we're doing these sort of custom therapies, right? But if I can just maybe ask the opposite, question. I think the thing that's so intriguing about a lot of genetic medicine, especially CRISPR, is you can envision why a therapy should work. Like the mechanism and the rationale is so clear. But I think as we know with medicine and drugs and just human biology in general, there's always things that surprise us. And we've really only tested, lipid nanoparticle delivered CRISPR medicines in a very small number, probably that I can count on one hand of diseases and genes, right? Some of those trials have had dozens, even a couple hundred patients, but it's still a pretty small group of people in the world that have been CRISPR'd per se. So I just wonder, like, are we getting ahead of ourselves here and thinking about, going from the big clinical trials where everyone gets the same therapy to these custom treatments? Is there a risk that this backfires?
Well, I think you have to take the data at face value, right? You've noted that only several hundred patients in aggregate have received lipid nanoparticle gene editing therapies, in vivo therapies up until this point. But you look at the results and they've actually been quite remarkable, right? So you think about most drugs, like take cancer drugs, like you're very happy if, you get, partial responses in some of the patients, and in aggregate, you get, a few months, increase in life expectancy, but you're certainly not expecting 100% of the patients to respond well, or even most of them to respond well. You're trying to, eke out feet in yards instead of going for the touchdown, right? Whereas what we have seen in the clinical trials with these LNP-mediated gene editing drugs are remarkable results. So, Intellia is very much in the news this week because they had a serious adverse event with one of the hundreds of patients they've treated so far. And, that's a subject I could expound on exactly what I think is happening and why actually I'm not concerned about the LNP being the issue. I think it's actually the gene itself that's at issue, but I don't want to get us off track. But what's notable is how almost all of those patients have responded beautifully to that treatment in terms of their transthyretin level, which is the target protein, coming down almost universally 90%, which unequivocally is going to help improve those patients' well-being. And it extends to other diseases. Intellia's other ongoing trials with hereditary angioedema, you see the same sort of pharmacodynamic effects over and over again with all of the patients who are receiving drug. These are not partial responses. These are not some patients respond, others don't. Across the board, we're seeing this. And in hypercholesterolemia trials, Verve Therapeutics and others, seeing the same sort of effects. Like these are very, very effective drugs. And so even though it's only a few hundred, the responses have been so positive. It's hard not to come away thinking, this is the real deal. And yes, unexpected things are going to happen. There are going to be adverse effects. No drug is 100% safe. But you don't want to throw out the baby with the bathwater, right? You want to take them at face value and say, okay, these are very good drugs. Let's figure out how to mitigate even those, very unusual side effects that can sometimes occur. And you do that by doing more clinical trials and revising protocols, and then,
Thinking
moving
about
step by step
these
towards
as
payments.
as drugs or as like a type of surgery or a medical procedure? I mean, it feels like drugs is kind of the obvious pathway that these go through, but like, is that the right way of thinking about this as a process and as a technology, and like what the patients actually look like when we think about what you have to do on a regulatory basis and on a product basis and on a commercial basis as we think forward in the years?
Yeah, I think that we have to work in the system that we have now while trying to still push the system in a way that will adapt it to the needs of our patients. And so right now we're sort of making incremental changes, or what seems to be even more from what you were just talking about than incremental changes, with the way that rare disease gene editing therapies are sort of regulated and looked at in this country. I think ultimately moving towards a genetic surgery model might allow us to scale more quickly and have access for a wider number of patients. And if you think about this sort of analogous to like an interventional cardiologist or an interventional radiologist, right, they are adapting what they're doing for the patient in front of them using tools. In this case, our tools are, our gene editing tools that are being developed by our manufacturing partners. And so I hope that someday we do get to a point where we essentially have personalized genome editing centers that can be credentialed and monitored and make sure that this is done in an appropriate and safe way, but would allow for the degree of flexibility that's needed to essentially meet the need of any patient.
I know we are running a little bit out of time. Ryan, did you have one last question we can get in before we let Kiran and Becca go?
I could probably think of dozens,
Yeah.
but let me end with one. It was a really remarkable moment when we saw baby KJ graduate from Children's Hospital this summer and finally go home with his family. How's he doing? Are there any updates you can share?
Yeah, he's doing great, right? He's
Yeah.
enjoyed his summer at home. He continues to grow and develop beautifully. And his markers, his biochemical markers of disease that we look at continue to look wonderful. And so we are making incremental progress in how we're managing him. That being said, I mean, I'm always nervous. I'm always going to be nervous, right? We're going to watch him very carefully, and we're by no means claiming that we have definitively cured him. But I think with each passing day, we are seeing that we at least made his really severe disease more mild. And I think I am being more convinced of that every single day, even though I'm the one that's the most nervous to say that out loud.
Well, I think that's a fantastic place to leave this. Thank you both enormously for being on the show. Congratulations on your work, the paper out today. Cool to have a little bit of live news as well, which seems like good news for what you all are doing. We hope to have you back and learn more about the work. So Kiran and Becca, thank you so much for being with us. That is all the time we have. Thank you, everybody in our audience, for joining us. And we will be following along with all of this closely, and you can keep up with our coverage on endpoints.news and by subscribing to our newsletters. The recording of this conversation will be on YouTube. We'll also live on Spotify and Apple Podcasts. If you've already signed up for our Post-Hoc newsletter, you'll get notifications when we have a new broadcast coming up. And if you are not signed up, head over to endpoints.news. Then in the top little right-hand corner, hit the reader profile link, then preferences, and you can activate Post-Hoc. You can also follow us on X, formerly known as Twitter, LinkedIn, YouTube, Spotify, and Apple Podcasts, and Blue Sky by searching for Endpoints News. Huge thank you to our producer, Nicole Wetzman, and our engineer, Nat Gonzalez. We will see you the next time news breaks. I am Drew Armstrong for Post-Hoc Live.